My name is Viktor Fursov. I am a research entomologist, beekeeper and teacher. And today I decided to make a stream about my subject of research about chalcid wasps. I did different videos devoted to different insects and they're uploaded on my YouTube channel. But today I will be talking about tiny chalcid wasps. Because some people who is interested in entomology and in microscopy, in photography, have asked me about how do you collect small parasitic wasps. Yes, I'm collecting different micro-hymenoptera. So they're very tiny. I'm actually studying very tiny parasitic wasps of the family Trichogrammatidae. And they're very small. They're here in this picture, in this album, big one. But originally, this is a Trichogramma and Trichogrammatidae family, egg parasitoids of beetles, butterflies, moths, and some sawflies and some other insects. They're very tiny, half of millimeter. But what about methods of collecting? How to collect insects, how to collect chalcid wasps? Because I'm specializing on chalcid wasps, superfamily Chalcidoidea, which is different from Ichneumonidae and Betyloidae and others. Chalcid wasps are pretty small, from 0.5 millimeter and smaller, up to maybe 1 millimeter, big one, and 5 millimeters, 6 millimeters, very big one, and a little bit more in some other families. But how to collect them? For photography and also for entomological research and study under the microscope, very simple one, for sorting, for preliminary identification, and maybe for making some microscopic slides like that. I'm doing tiny microscopic slides. I'm putting my trichogramma on small micro slides. I'm making preparations to watch them under the 1000 magnification under the optical microscope Olympus with very high magnification, 1000. So to watch for structure of morphology, antennae, heads, abdomen, ovipositor and male genitalia of these tiny parasitic wasps. But today I want to tell you some simple methods how to collect them, how to find them, how to take photo of them under the microscope, if you found them already, or just to take photo of them in nature, or maybe in laboratory, carefully rear some material, living material, and it's quite interesting to observe for chalcid wasps, because they are very interesting. We have a book devoted to chalcid wasp, Atlas of Entomophagous Insects, of European part. In Europe, especially this is devoted to Ukraine, with different entomophagous insects from different superfamilies. These are not photographs. These are white and black and just colorful pictures. Colorful pictures of a family Braconidae. This one, family Ichneumonidae. Different entomophagous insects. Some wasps, some ants, even some interesting tiny Incertidae family, chalcid wasps, and even some tiny Aphelinid wasps, Telenomus, proctotropoid wasp, and some very funny Tachinidae parasitic flies. So many of them can be reared from hosts. But not only by rearing. There are two most important methods of collecting. First of all, collecting three methods, by the way. One method with entomological sweeping net, not like that. This is aquarium net, very tiny one. Or another one, which can be used for second method. I have very big entomological net, 60 centimeters in diameter, so you can sweep with butterfly net through bushes, through grasses, just beating just some leaves on trees, on shrubs, and some tiny, very tiny parasitic wasp up to 1 millimeter, half millimeter falling down with leaf litter on the bottom of your entomological net or butterfly net. So, and you can collect them with putter, with aspirator very carefully. But for that you need to have very sharp eye, and some of the insects fall down and you cannot find them because they're very tiny. So because they escape, they fall down and they're smashed. They can be destroyed by leaf litter. For that, so this is the first way where you can be very precise to find precise species on precise plants in a bush, sweeping by entomological sweeping net. This is very nice, especially for bigger insects, up to maybe 6 millimeter size, big chalcid wasp and some braconid wasp and some ichneumoniid wasp up to 15 millimeter size. Entomological net is unreplaceable stuff for entomologists and for hymenopterists. Second method, also very effective. I have told about it already in my many videos, but I need to repeat it again and again because we need to use this very tiny stream technology to distribute the information how to collect insects, how to study them. This is method to use yellow pen traps or yellow dishes or yellow bowls. How to use them, but to collect already dead material. So because living insects will be flying around and you can fall down into this yellow pots, yellow plates, yellow dishes, yellow bowls, and die inside. So you need to use water, clean water, fill the bowl with water, add to water 1 spoon of salt for 1 bottle like that, for 1 liter. Mix it and add also 1 tiny small teaspoon of detergent or shampoo. Very easy one, but better without smell. Better without smell, lemon smell or other lavender smell. No, it's not. Lavender smell not very good. Sometimes it's not attracting insects, but disorientating them. So fill water. Here maybe half a liter. Then water already with detergent, with salt for preservation. Fill with water and put it on a grass 1 by 1, maybe on distance 50 centimeters, 1 centimeter. At least you can watch them, so do not miss them when you can come back and collect them. Sometimes you can use hard plates with very good plastic, or sometimes you can use one-time used plastic, very tiny one, which can be easily destroyed, but very cheap. So very cheap. Size is not important. This is the perfect size, about 15 centimeter size. Can be smaller. It's up to you which bowls you can buy. And sometimes they're not so deep. This is optimal, maybe 3 centimeters deep and 15 centimeters in diameter. Fill with water, with salt, with shampoo, and put it just better for 1 night. For 1 night or half of a day and 1 night, and then collect it next morning. Because if you put them in springtime and summertime, water will be really hot here. And insects sometimes swarming and can be destroyed because of hot water. But they can be preserved if you add here some salt or maybe some glycerin as well for preservation. After that, after spreading just on a certain place with vegetation, with flowers, under the flowers, between stones, under trees. Sometimes you can put it even on tree, attach it somewhere just on branches carefully, or just on stone near the river, just on salty sand, or just near the salt, or on the sand near the lake, near the pond, on grassland, whatever you want to find. Insects, whatever you search. Insects are flying around like plankton very quickly and fell down, probably because of yellow plates. Yellow color because most of Hymenoptera, micro-Hymenoptera, and just macro-Hymenoptera like bees and bumblebees maybe too. Some bees and some others, insects, fell down and were dying inside salted water. But next morning you are coming with tiny aquarium net. Tiny aquarium net like that. I use this one, small one, which is very convenient, flexible, and very tiny mesh. And I fill this tiny net with water, put water with all insects inside. Sometimes can be half of this mesh, half of this net can be with insects. One plate, then I go to another one, put another plate, go to another one, put another plate. Fill my net with insects, and then I do not clean it immediately in field conditions because it's hot, it's summer, you know, it is field. So I put it somewhere just in a bag and bring it to the laboratory, to a field laboratory for sorting and for cleaning. Just in laboratory, all insects must be cleaned, or in field conditions, again with a clean water to take off detergent from here, because detergent will make just white cover around the surface of insects. So you clean your insects under the water, clean water, just like that. Insects are very flexible, they are not destroying. And then after that, you can take them off again and put them to tiny, I prefer these bottles, this stuff of bottles, to make not very big samples, to make relatively comparable samples. So I put them inside again, from water, clean water, to alcohol, ethanol. 70% ethanol or 90% ethanol. It will be less because it will be a little bit water inside insects. So, and you can fill this bottle with, with children's food usually, or some spices, or with some yogurts, up to the top or just middle. So insects are flexible, so they will be keeping here some alcohol, and they will be preserved for ages, for those years. And put here again label, very important part. Put label, so this material will be very resistible for damage, very clean, because you cleaned already in water, and you can use it yourself for sorting under the microscope, for sorting for families, for species, for genera, and then for species. And then you can use this material fixed in alcohol for microscope slides, for preparations, and for identification. Okay, we understood about collecting some dead insects in yellow pen traps, or we call it in Malay trap, using these small aquarium nets with very tiny mesh for sorting. Yes, but here you can collect dead insects, but very tiny. Sometimes many other not interesting insects coming here. Sometimes flies coming here. Some snails crawling inside. Some mice can come and die here. So you need to you have to spoon or just by stick, take them off. If some insects are unimportant for you are coming here, maybe just full of flies. Sometimes very difficult to sort them. If they are too big, or like maybe some big grasshoppers coming here and they are not important. You can, or snails. Snails making here just some soap inside, which can destroy other insects. Or some butterflies coming. Butterflies already not used for identification. You take them off. And fill the net with your sample. So the next day you check these plates again. So you understood a little bit, I hope so, about yellow pen traps or Malay traps. Not Malays. Malay trap, this is a big trap. Mercury trap or yellow pen traps. Second way for collecting, ermigines. This is emergence from hosts. Hosts can be insects, arthropods, and plants. There are some plants where some insects, phytophagous insects, are living inside the roots, inside stems, inside flowers, inside flower pots, and inside seeds. And also inside some damages of plants, like some galls, some swollen places on plants. So, and many insects are making galls. We have 3 books published in our department devoted to gall insects, which are making galls. So one book, for instance, here, insects, gall makers. This part of Diptera. There are also second part devoted to Hymenoptera, and third part devoted to Coleoptera, Lepidoptera, and some others orders of insects which are making galls as gall makers. Galls are quite visible because some leaves, some stems are damaged, some pods. So you need to observe carefully plants, and you can find some plants are damaged. So you can collect some pieces of plants inside you, put them in plastic jars or in plastic packages, and you can observe with filter paper and carefully observe for these insects, for this sample. So some phytophagous insects can emerge from these samples with galls. And especially now, this is autumn time. In autumn time, majority of insects, especially in temperate regions, already preparing for overwintering. So that's why all plants, all stems already are dried. So now you can collect dried galls, which are prepared for overwintering. So you can collect some swollen pieces of plants, which in many cases are done. So you see here, galls can be extraordinarily different. This is an oak tree, which are making some centipede. Wasps are making a lot of different galls. Oak tree, extraordinary tree, with a lot of different galls on oak tree. And many other families of trees as well. They're making very funny, very interesting galls, such unusual pieces of plants. Inside these pieces, inside this gall, different insects are developing, as I said, Hymenoptera, Diptera, Coleoptera, Lepidoptera, and others. So you can collect galls, you can collect this piece of plant, put them in a plastic jar, like that. Plastic bottle, in a glass bottle, maybe bigger one than this one, maybe half a liter, maybe 1 liter. And you can wait till the beginning of winter or maybe middle of winter, because many insects are hibernating, but in warm temperature in laboratory, they are preparing for emergence very quickly. So some of them very need diapause, but some of them don't need diapause, and in warm temperature in laboratory, they will emerge. Some pieces of grasses as well. And so making some small galls, bigger one, on pods, on stems, sometimes on roots. These are galls. If you can find some unusual, like a disease on plant, disease on fruit, disease on leaves, this can be gall. Damaged, destroyed, deformation of stem, deformation of pods, deformation of leaves, and you can observe them. So you can receive phytophagous insects which are developing inside these galls, and after that you can carefully observe how they are developing. So you can receive also parasitic wasps of a family Chalcidoideae, chalcid wasps, and maybe some platygastridae wasps, maybe some braconid wasps. It's up to the size of galls, it's up to the size of host, which insect is developing inside these galls. So you are keeping sample in plastic packages with zip, for instance, and wait for few months. Definitely you need to put inside filter paper to wait, and you can receive absolutely valuable material of phytophagous gall makers, gall feeders, and parasitic wasps which are developing on gall makers. So you can receive precisely host, which is developing on precise plant, and you can receive parasitoid, which is developing precisely on this phytophagous insect. And this is very valuable scientific material which can be used for publication. An entomologist, they have an obligation to do publications. So you can publish it, and you can find very interesting material, especially if you're living somewhere in distant area in the country where nobody didn't study it. If somebody do not understand me, learn English. I'm speaking English because English language is international language. So if somebody from not English speaking countries watching us, welcome to watch my other videos where I can speak Russian and Ukrainian language. Today I'm speaking for English speaking audience, and you know, now all countries using English language. And entomologists and scientists and biologists are obliged, where necessary, to use English language for interpersonal communication, for communication between amateurs and scientists, between professionals and between amateurs, and to develop even also citizen science. So maybe this video also for citizen science, because majority of professionals keep their methods in the secret, or they do not talk about them in outreach activities. They do not make outreach activity. They are publishing their very strict scientific papers and saying, you can read it in the paper, you can download it from ResearchGate and study it, and you can use it. Everything is described because it needs time, efforts, interest, and enthusiasm to explain about it. So I'm entomologist, as some people enjoy. Hello, Stas, who is our visitor, and Katerina maybe just somewhere in China. So some people are visiting us from China, some people are visiting us from other countries. So if you are from English-speaking country, or you can understand me what I'm talking about, write in comments what is your home country. Yes, how to get biology. The biology, this is the most important. I'm making this stream from my laboratory, from our laboratory of our Department of Entomologist Insects and Ecological Principles of Biological Control, from the center of Kyiv, capital of Ukraine, and I'm located near Metro Teatralna, Bogdan Khmelnytsky Street 15. So you can visit me if you are coming to Kyiv for traveling, for curiosity, or for scientific purpose. We can discuss in person some scientific questions and outreach activity questions. Today I'm talking about methods of collecting parasitic wasps, small tiny chalcid wasps. First part, to collect different galls. Galls, these are different modifications of leaves, stems, leaf buds, sometimes bark, roots, and other parts of seeds, flowers, which are making such kind of deformations, and inside some insects are living. Cut piece of plant, put it in plastic jar, in a plastic package, in big plastic jar like that, or smaller one, or put it in a emergence traps. Search my other videos about emergence traps. This is tissue bag with attached glass box. So insects are coming from tissue bag to the light. And after Gulls, I want to tell you this book devoted to gull workers, gull feeders from the order Diptera, as I said. Some others, we have 2 books more published in our department devoted to Hymenoptera, devoted to Coleoptera beetles, and devoted to Lepidoptera butterflies and moths. But I wanted to tell you more about other parts of collecting insects using emergence traps from different seeds, from different pods of Fabaceae, from different beans, for instance. Katerina is watching us from China, maybe not from China, I guess. Ah, yes, you are from Ukraine. Very good, very good. So do you know Chinese? It's lovely. Northern Chinese agricultural bugs. Yes, Chinese books I keep here. I visited China. Be careful. Oh, we're talking about Chinese bugs. These are Chinese agricultural bugs. So I keep them. This is a gift for my Chinese colleagues. Yeah. Here are some damages of plants and life cycle of different insects, with description of life cycle and with quite nice photos. Because some insects, they have a wide distribution, not only in Asia, but also in, we say, in Palearctic region, and some species are introduced to other places. So many southeastern insects, like this, different night moths, are widely distributed globally. They're becoming invasive species, including in Ukraine. Like now some Southeast Asian brown marmorated bug is coming to different areas of Ukraine, and I collected this brown marmorated bug in Kyiv, but 5 years ago it was only expected as a quarantine species. But now many of them flying around in our botanical garden in the center of Kyiv. So that kind of a more or less outreach books, not only scientific but also with some popular photographs, but can be used for identification of damages insects, damages of plants, and identification of eggs, larvae, pupae, and adults of different insects. So this Chinese book, but we have also just our Ukrainian book with parasitic wasps, as I said. With parasitic wasps, with some description, this text about life cycle biology, and also some very nice colorful photographs, colorful pictures of ichneumoniid wasps, parasitic wasps, ichneumanoid wasp of superfamily Ichneumonidae, some braconid wasp family, Braconidae. So they are all done by our devoted artist, biologist, who's drawing them. I've also participated in drawing it. I used all samples myself. I drawn all details here before final coloration of all insects here. All tiny details of wings, segments, antenna segments. I did it myself. But unfortunately I am not an author. I was deleted at the final moment by editorial committee, surprisingly. But I did a lot of pictures here myself. And here, this is a drawing of family Chalcid, the superfamily Chalcid wasp, family Ensortidae. This is family Eolophidae, family Aphilinidae, parasitoids of sucking insects, and some tachinid flies, parasitic flies. Wonderful very careful, very precise scientific pictures in white and black. And this is my subject of research here. Tiny trichogramma. This one, 0.5 millimeter size insect, which I'm doing usually for study, making some microscopic slides for identification. So, this story about books was about pods. Finally, I want to tell you a little bit more about rearing from seeds, from fruits and seeds. So you can collect different seeds in glass jars or in plastic jars like that. Here, for instance, some seeds of malva, mallow tree, or this pod of lydicia tree, big one. It's already with some open exit holes, which were done by seed beetles, Megabrochidius dorsalis. And for instance, here some seeds of linaria I collected today. Some tiny weevils of a family, Curculionidae, are developing on seeds. So if you collect some seeds in plastic jars, okay, without holes, definitely. Close with cotton here for evaporation, because otherwise all insects will be just with fungi. This is temporary fixed here. So it will be evaporating, moisture will be evaporating slowly, insects will be developing slowly inside, and plants are drying slowly, and you can observe it after maybe couple months or maybe 3-4 months, even in the winter. Some insects, which are seed-feeding insects, will be hatching down out from seeds, from this one and from this one. Maybe some of them can come out immediately if they're hiding somewhere. But if you didn't collect them, they will hatch very soon. Maybe in period 3-4 months, especially they can come usually before springtime. And as an example, I want to show you how many of them. It's a lot of different species have been collected in our department of entomophagous insects and ecological principles of biological control in Kyiv. This book was written by two authors, Professor Zerova and Dr. Seregina, Seed-feeding chalcid wasps. Seed-feeding chalcid wasps from 3 families, mostly Torimidae, Ormiridae, and Pteromalidae. Here indication of their host-trophical relations, and also pictures, only in this time was white and black, very careful scientific pictures, drawings of chalcid wasps, female and male, female with very long ovipositor, because chalcid wasps, which are seed-feeding insects, they are searching for seeds inside fruits, for instance, inside some places, and they need to use very long ovipositor for ovipositioning inside seeds, and it's hard job. And for sure, to be precise about life cycle, some seeds can be open, and if you are opening some seeds, you can be sure that this is larva of seed-feeding insect, larva of chalcid wasp, or maybe some larvae of other insects like beetles. So you can identify beetle, or you can identify this is a chalcid wasp, to be precise in some samples. And after that, after so precise identification of larva and pupa, so you can identify carefully adult. Here are different pictures here with indication here. For instance, here family Eurytamidae with some indication of seeds on between insects, tiny pictures of seeds. Because seeds are making with holes. These holes are emergent holes. Because these tiny insects with size from 1 millimeter up to 5 millimeters, they're hatching from the seeds, and it's hard job. They're making a rounded exit hole, And they are very specific for different species. And you can identify, because some insects are emerging even from very tiny seeds. Maybe seed is 3 millimeter size, flattened, not very big one. And especially belonging to different families, you can, for example, I can read here a little bit about families. You can be surprised how many families are used for seed-feeding insects. So here, for instance, distribution of chalcid wasp for families of different host plants. This is taxodiaceae, pinnaceae, cupressaceae, ephederaceae, hamamelidaceae, aquifoliaceae, anacardiaceae, rosaceae, brassicaceae, euphorbia, scrofulariaceae, apiaceae, lamiaceae, liliaceae, poaceae. All these different families were attacked by phytophagous chalcid wasps of different families. So we're seed-feeding phytophagous insects. Here even very special plate, table with indication. Plant, species of phytophagous insect, chalcid wasp. Species of plant and distribution in the Holarctic region and Palearctic region globally, worldwide. Don't forget to write in comments what is your home country, where are you watching us. So some people are watching us from Belarus, some people from Ukraine, maybe some people from USA too. And this, my story about seed-feeding insects, is devoted to some amateurs, some professionals, some photographers who is interested to collect living material of insects. For that you need to collect some seeds of different plants. Are you going to excursion to the park, going to the forest, going to the river, to pond, and you can see here in autumn time different seeds. Collect them in a plastic jar, collect them in a plastic bottle, collect them in a tissue package, in plastic package. And this will be just emergence package, because all these insects are emerged from different seeds, sometimes like this one, seeds inside fruits. Some dry fruits, like this species of Eurytoma, by developing inside some seeds on fruits, like amygdalus seeds, or some in seeds of rosa are developing some species of Megastigmus genus. So far so good, quite easy to collect seeds. You keep them, and in mid of winter or in late winter before spring, many tiny insects, chalcid wasp, will be emerging in your jars, in your emergence traps. So you can use them for fixation, for scientific purpose. You can fixate them in small tiny Appendorf tubes in alcohol or dry. Dry is perfect. Put dry insects inside these Appendorf tubes, very tiny one. Appendorf or any kind of tubes, or this tiny Appendorf tubes, plastic. You can put them dry, or you can put them in an alcohol. Dry is even better. Put dry insects inside and put here just a piece of cotton for fixation. Put here inside label with description of country, city, village, date of collection, date of emergence, collector, and some details. Plant, very important. Fix it dry. You can put bigger label outside, fix it with scotch. So, and keep it, and you can send it for identification to scientific laboratory, like to our laboratory in Kyiv, capital of Ukraine, on address Kyiv, Bogdan Khmelnytsky Street, 15. Department of Entomophagus, Insects and Ecological Principles of Biological Control. We have team of biologists, entomologists. We are studying different families, and maybe some insects can be new for science. They can be identified, or maybe they can be described as new. If they have not been found before, if they have not been described before, they can be not only new in distribution, insects, but also new for science. It means they never been found before. If you are living in some distant area, or not in distant area, but in some a little bit unstudied area like Tajikistan, Kazakhstan, Kyrgyzstan, Uzbekistan, Indonesia, Vietnam, Cambodia, Thailand, Philippines, India, Sri Lanka, Australia, African countries, Tanzania, Kenya, Somalia, maybe Angola, maybe Mozambique, maybe Tanzania or Niger. Or maybe you're living somewhere in Europe, in temperate region, somewhere in Portugal and Spain. You can collect also some insects in temperate region, or maybe you can collect them in tropics in Brazil or in Central America, somewhere in Guatemala, where people are just immigrating everywhere, all around the world. But maybe some devoted naturalist can be interested in identification of insects, like in Panama. Panama is pretty well-developed countries. I know some entomologists from them. I contact them. And even I described some species of new species of Trichogramma from Panama. Some devoted and famous Dutch entomologists visited Panama, studied some butterflies, collected eggs of butterflies, Heliconine, and they collected from eggs of butterflies, Heliconine, egg parasitoids of genus Trichogramma. And together we described a couple of years ago 3 species of Trichogramma genus. And even we got some award from British Museum of Natural History, but still we have not seen this award a few years ago because of this COVID and so on, so all these problems. Okay, hello to Hungary. Beatblaze, hello. I didn't know what is your home country. So far, so good. Try to collect some seeds. You can collect them in different jars, like I collected here. Number of seeds is actually difficult to count. Everywhere you go to a forest, everywhere you go to meadow, you can find some seeds, like these acorns. Even inside these acorns, there are some phytophagous weevils. I'm not quite sure that there are some seed-feeding chalcid wasps, no, but some phytophagous curculio are developing inside and they're hibernating in soil now. But the same way you can collect here inside different seeds or different fruits like rosa. Keep them inside and can watch what is inside. After 2, 3, 4 months, some insects will be hatching and you can collect them carefully, as I explained, inside, up indoors. So you can send them to our department for identification, maybe for description of new species and for collaborative publication if you are interested in scientific research, in scientific study, and if you are interested in cooperation. So if you are somewhere in Cambodia, Vietnam, Thailand, Indonesia or other countries, maybe in Ecuador, or in Peru, in Guatemala, in Venezuela, or Argentina. Welcome for cooperation. So don't forget about funny, interesting pods of Fabaceae, like this one, Lidicia triacanthus. In some countries close to Ukraine, you can collect Lidicia triacanthus in plastic boxes, so you can rear some beetles and maybe some entomophagous insects, which are developing own beetles as well. So this method is called emergence traps, and this method is called rearing from seeds, because today I wanted to introduce you to this book, Seed Feeding Insects of Chalcid wasps, seed feeding insects of a family Torimidae, Oromeridae, and Pteromalidae, superfamily Chalcid wasps, Chalcidoidae. This is a story about parasitoids. This is a story about phytophagous insects. This is a story about seed-feeding insects. Some not English-speaking audience can write in Russian and Ukrainian. Можете написать, если вы нас смотрите из Украины, из какого вы города нас смотрите. Катерина нас смотрит из Украины. Сегодня мы рассказываем о хальцидоидных особях, истцах, наездниках, которые паразитируют на различных насекомых, в том числе паразитируют на различных галообразователях, потому что есть насекомые, которые фитофаги, живут как фитофаги в галах, а на них живут паразитоиды, энтомофаги. А здесь есть еще фитофаги, живущие на семенах. Можно собрать различных насекомых на семенах, содержать их в пластиковых пакетах, в пластиковых коробочках, в полиэтиленовых пакетах, зип-пакетах, вот таких зип-пакетах, как с Эппендорф я показываю. Эппендорф закрывается на зип. Плотно закрывающийся. Можно сюда положить фильтровальную бумагу, белую, чтобы было видно. И сюда вот такое же количество положить различных семян, самых различных растений. И обязательно выведутся фитофаги, семиеды, которые могут быть интересны по распространению. Может быть, они никогда раньше не находились в этом месте. А может быть, даже будут новые для науки виды, которые можно будет определить и использовать для публикации в научной литературе энтомологических научных журналах. Будет пример для кооперации, сотрудничества в деле развития энтомологии. Today I was doing this story about methods of collecting entomophagous and phytophagous insects of superfamily Chalcididae from seeds, fruits, stems, galls, flower pods, and other places of insects, plants, from plants and from insects as well. So you try to use these methods to breed, to rear insects from using emergence traps, especially using emergence traps using seeds. So the seed-feeding insects of family Oromeridae and Torimidae and Pteromalidae, Chalcid wasps. Try to collect seed-feeding insects, and it will be very interesting, unique material for study, for identification, and for scientific publication. So, and you can receive different interesting, sometimes even big insects, like ichneumoniid wasps, and in many cases smaller braconid wasps, parasitic wasps, and in many cases very tiny small phytophagous seed-feeding insects, like this family Eurythmidae. And if you are interested in cooperation, in collaboration, in identification, try to visit iNaturalist, and try to use me and my colleagues in Kyiv, capital of Ukraine, in Institute of Zoology of National Academy of Sciences. You can find my address in the description of this video. And don't forget to subscribe to my channel if you have not been subscribed yet. Have you subscribed? Not yet? Try to subscribe. Some insects are flying around, escape from my jars. I need to take them. So thank you for coming. And I want to say, don't forget, stand with Ukraine, support Ukraine, and help to Ukraine. Thank you for subscription. If somebody subscribed, thank you for subscription. Don't forget to subscribe and visit my Patreon page for some donations to become sponsor. That will be very nice. Sometimes if you are a sponsor, your message is immediately visible here in chat as a green message of sponsor. You can check Patreon page and you can check bottom of sponsorship. If you are living somewhere in Africa, Middle Asian countries, somewhere in India, Sri Lanka and Thailand, Cambodia, Indonesia, or maybe in lovely Australia, or maybe in very hot South America, in very hot South Africa, welcome for cooperation. Over in very hot Algeria as well, pretty hot place with unique insects in green places and a lot of different seeds and fruits like amygdalus are parasitized by Eurytoma maslovsky, Eurytoma samsonovi, and maybe some unstudied species of seed-feeding Eurytoma genus phytophagous seed feeders. This is also very interesting subject for the study. Thank you for cooperation. Stand with Ukraine, support Ukraine, help Ukraine, and Ukraine will win. Thank you for interest and curiosity. I hope something was useful, surprising, and interesting for you. Don't forget about seeds in your area, somewhere in Mexico, somewhere in Guatemala, somewhere in Venezuela. Some people have immigrated already to Ecuador, Algeria, everywhere, even to Nigeria, even to South Africa, to escape from difficult places, by the way. Don't forget that we are now in full-scale war in Ukraine, between Russia and Ukraine, so we have sometimes air alert sirens, so I need to be in a hurry to go to my underground station to take my train, because if there is a air alert siren, metro is stopped. I need to wait for next message in smartphone, but I can go. And sometimes it's long time. So thank you for watching. Press like. See you soon on my channel. Thank you, Bali, for appreciation, for your curiosity, and for support for this. Your message is a feedback that something was useful, interesting, and surprising. And it's important that if you rear some insects, you can take photos of many insects. Take photos, because insects will be crawling and they're alive, and they're sitting inside box. They're sitting inside jars. They're not going to field. You're not going to jungles. You have a lot of different insects inside your jars. Absolutely wonderful material for taking one nice photographing. A lot of photos can be used. And also this material can be used for scientific cooperation. You have questions, send me message, send me photos for identification. It will be kind of cooperation and outreach activity to spread knowledge about entomology, about insects for public. Thank you for interest, curiosity. See you soon on my channel and in a new stream. Good luck. Best greetings from Kyiv, the capital of Ukraine. This was Viktor Fursov, entomologist, beekeeper, teacher. Subscribe to my channel. Don't forget about it. Good luck. Thank you for visiting my stream.